Hello guys, welcome back to another GCSE video. Today we're doing a 10-minute test on key concepts in biology. Hopefully you can read all the text alright, so if you want you can just pause it here and try and have a go at these. I'm going to do it now I'm only going to give myself 10 minutes to do this whole page, and at the end I'll go through the mark scheme. So with that, I think I'm all done. 2 minutes to spare as well, so yeah. I think I took a little longer on the questions than I normally do, but I'm guessing that's just because I haven't really done any biology today. But yeah, so let's see how many marks. So does it tell me where the mark scheme is? Hang on. I'm guessing it does say where the mark scheme is. Oh, the answers are at the back. Alright. Okay, so we should have got A. Yep, we got A. The bacteria cells don't have nuclei. They have plasmids, which contain DNA. I think they're like rings of DNA. Or it might be they have a single strand of DNA with plasmids. I'm not. I think the plasmids are the strands. I'm not entirely sure. B. You can Google that, by the way, if you want and you don't know. B. We got that right. They moved to an area of higher concentration to an area of lower concentration because they want to spread out and make the concentration even. Chloroplasts, C, absorb light energy to make glucose. The reason chloroplasts are only found in plants is because they are used for photosynthesis, so they absorb the light energy to make glucose, which feeds the plant. For A, if the concentration of water inside a cell is lower than outside the cell, the net movement of water particles would be into the cell, because as we said earlier, it moves from an area of higher concentration to an area of lower concentration, and if the inside of that cell is the area of lower concentration because there's less water, then more water is going to move into there to even it out. 5. I'm not very good at microscopes. I might have got this one wrong, but I don't think I did. 5A. Yeah, you start with the lowest powered objective lens when you first use the microscope, and then you focus it to find the best resolution or something. To be honest, I don't really know a lot about microscopes. I just remember seeing this on Seneca once, so I guess it paid off. So at 37 degrees C, it took Amy Lay's 80 seconds to break down all the starch in a solution. What was the rate of reaction? I couldn't remember the formula for this one, so this one was kind of a guess for me. I think I remembered it being a fraction with the thing at the bottom though. So I realised 37 isn't part of the rate of reaction, because the rate of reaction is just a measurement of how much reaction happened in a certain amount of time. So I took that out of it and just put 80, just did 1 over 80, and I think that's 12.5. I had to find my calculator for this, but I don't think I used it in the end. I just kind of took a guess. So that is 6C. Yeah, so I did get it right. Yep, so the equation must be 1 over 80 then. Times 100. Yeah. So 7. Why Yeah, because the best thing to do for this is you can rule out the temperature's not involved, so it's just this number. So you've got to find a number that's somehow similar to 80. Like 8, if you're not sure, if you're just guessing, like 80 divided by 4, that's not going to be the equation realistically. It's going to be like something squared or square rooted, so you just want to find something similar. Like, I just knew that 1 over 8 was, I think, 12.5. So I saw that and I was like, oh, it must be that one. 7. Why is the shape of an enzyme important for its function? C. So that it fits the substance involved in the reaction it's catalyzing. So it kind of fits the substance. It's how it works is the enzyme has an active site on it, which is like a certain shape, right? Which is like a certain shape, right? And the I think it's called a substrate. The other one has an active site that's a certain shape, right? And it's got to fit in that certain shape. You see, right? And that forms an ESC, a enzyme substrate compound, and that lets the enzyme break down the thing, right? 8. Proteases catalyze the breakdown of B, proteins into amino acids. So I got this one right. You can tell because there's a clue in the name. It's protease. Normally with these, they sound a bit like the thing they're breaking down. Like protease sounds like proteins, but they've just changed the ending. Like for lipids, it's lipase, and for carbohydrates, I think it's carbohydrase. Yeah. So there's often a pattern. Not for all of them, I don't think. I think there might be like 1 or 2 where that doesn't apply, but for most of them, the enzyme has in the name what it breaks down. 9. Explain what's meant by an enzyme being denatured. The shape of the enzyme's active site has changed, so the substrate won't fit in it anymore. Well, I didn't quite write that, but I wrote something similar enough, I think. An enzyme becomes denatured when the temperature is too hot and the active site deforms. That's what that's meant to say anyway. I don't know if you can read my handwriting. So, would that be right? I didn't mention the substrate, but I did say that the active site deformed, so it changed. So, I think that's a good enough answer. Yeah, I think I'd get the mark for that. It's only 1 mark anyway. 10. It becomes tough and rigid to avoid being penetrated by other sperm. That is what it says on the answer. And then it says on the mark scheme which makes sure that the offspring has the correct amount of DNA. To be honest, I didn't think that they'd put a second part on. I thought that it might just be it becomes tough and rigid, and then to avoid being penetrated by other sperm on the mark scheme. And I couldn't quite remember the word for what I was trying to say. But in the end, I remembered because there would be too many chromosomes that should really say multiple sperm, not just 2, because that means it's fine when there's 3, which it is not. It's probably much worse. So yeah, you wanted to have the second bit as well, where there would basically be too much DNA, because the 2 haploid cells are meant to combine. But you can't have 3 halves, because that messes up. You've got like 1.5 humans, and the nucleus doesn't really. When the DNA like makes the embryo, it doesn't really know what to do, so it will deform it a little bit. I think that would be quite a bad one. I think. So. Yeah, that would not be good. That's why it stops that from doing that. Complete the diagram of an animal cell. I was about to write cell wall, and then I actually read the question and saw it said animal cell, so I changed it to cell membrane, and I think that's a ribosome. I wasn't really sure on the dot. It might not be a ribosome. Yeah, it's a cell membrane and ribosome. Nice. Ribosome was kind of a guess. I didn't think they were shaped like that, but because it's just the only other thing I could think of that's in an animal cell. But yeah, no, it was right. You have the mitochondria, the cytoplasm, and the ribosome as the kind of subcellular bits, and the nucleus. But I mean, that's not really dotted around. There's just one. Describe the roles of the following part of the cell. Mitochondria. I honestly could not remember this, but this was the best answer I could think of. I think mitochondria lets the cell respirate, but I'm really not sure on that. I'm really not sure. Oh my god. It is my lucky day. Mitochondria are where most of the reactions for aerobic respiration take place. Okay, so really, rather than just saying it's something to do with respiration, I shouldn't really give myself the mark for that, because it's the reactions for aerobic respiration. So, because it can't be the anaerobic respiration, because that doesn't involve the mitochondria, and it has to be the actual reactions that are happening, like, can't remember what the formula is now, but it's that, the formula for respiration. And the nucleus contains the genetic material that controls the activities of the cell. I just wrote that the nucleus holds the genetic material of the cell. Don't know if I was really meant to write that controls. So yeah. To be honest, I don't know how much I should give myself for this one. To be honest, I'm going to be nice, because all of my answers, while they weren't always the best phrased, they did have the right answer, and I think I would be being too harsh to deny any of these marks, because, I mean, they're all true. They're just not, this one's just not in very much detail, so you would want to do more than that on the test. But I'm just going to write 15 out of 15. Yay, that's a good start. Hope you've enjoyed this 10-minute revision video.